Hi, my name is Josh Devere, and today I'll be presenting MagGenix. Every year, millions and millions of Americans undergo a dysregulation of their immune system that can lead to a variety of local inflammatory diseases. These can include things such as rheumatoid arthritis, organ rejection, cytokine release syndrome, and even cancer. Currently, treatments for this can include things such as CAR T therapy or monoclonal antibodies, such as IL-6 blockers, like tocilizumab. These can be somewhat effective, yet they both lack localization, meaning that any therapeutic response affects the entirety of the body and can lead to conditions such as systemic immunosuppression. This is such a hindrance to patient lifestyle and their health care. Thus, we developed a need statement that immunologists and oncologists need a minimally invasive and location-specific method to increase efficacy and reduce the side effects of immunomodulation. Thus, we've developed localized immunomodulation via magnetic control. Our platform allows for ex vivo engineering of B and T cells, as well as prokaryotic and eukaryotic organisms, with our novel plasmid DNA that allows for localized cellular activation and modulation. Utilizing the patient's own cells in analogous T cell infusion, we can give current Treg and CAR T cells a high degree of spatiotemporal resolution, meaning we can activate them specifically when and where we want them to. Looking at two examples, with brain cancer, we can utilize our technology to activate immune cells, such as CAR T cells, only in the region where we want them to function, such as the tumor site. Additionally, with immunosuppression, we can activate Treg cells to downregulate immune responses and inflammation only at arthritic joints. This means that we can have lower amounts of side effects while increasing the intensity of treatment at the site of affliction. We provide care to patients in this increased intensity of treatment. We provide value to providers in the control that we give them over their patient and their treatment scheme, and we provide value to payers as we can reduce the overall cost of treatment in a patient lifetime. Within a $1.5 billion T-cell engineering market with an astounding 39.1% compound annual growth rate, we stand out among the competition. Things such as small molecule immunomodulators, biologic immunomodulators, cell therapy immunomodulators, which despite a degree of immunomodulation, lack localized treatment that hinders the degree to which they can actually alter the immune system. Our technology operates through three main operation channels. Firstly, we act as a CDO, contract development organization, with biopharmaceutical or bioproduction companies. In other words, we can take their current CAR T therapy and, utilizing our proprietary software, engineer them to be magnetogenetic and receptive to magnetic and electromagnetic stimulation. We can also allow for licensure of our proprietary DNA and RNA cell therapy platform to these biopharmaceutical, bioproductive sources, as well as academia, where we can also allow for sale of off-the-shelf cells or organisms that are enabled with our magnetic stimulation. This provides the ability to target not only patient populations, bioreactor and other bioproductive services, and academia, where we can allow for research that replaces current optogenetic techniques. Milestones we've achieved include validation of our concept, in-house tooling with our proprietary software, funding of dry and wet lab endeavors, as well as development of our MagGenix MVP inside of E. coli samples, which we are currently in the process of characterizing. We're seeking $20,000 to allow for the development of human CAR T minimal viable prototypes, the launch of our nucleic acid design software, and lastly, the sale and licensure of our off-the-shelf MagGenix cells to academic institutions. With our team of 4 Hopkins undergraduate, 2 BME faculty advisors, we believe that we can bring magnetogenetics to the healthcare world in order to enable localized treatment in various patient populations. I've been Josh, we've been MagGenix, and thank you.